Okay, hello and welcome back um, to I guess we're on week 4 now, or week 3, but we're on chapter 4 of the textbook. If you're following along, the textbook's not required. But this week we're talking about the distributions of species and some of the ecological foundations that can influence the dynamics of the geographic range of where species is located. And something that's worth mentioning, every species is going to have its own unique geographic range, and it's going to have its own set of abiotic and biotic factors that influence its geographic range. And so, what we want to talk about is some of those things that are going to be, what are the underlying components or the underlying sort of interactions between a species and its environment and other species and other members of its own species. How do those all sort of culminate into a species' unique geographic range? And so I kind of want to start at the bottom here and kind of work our way up. And I want to look at some distributions of individuals, or at least talk about the idea of mapping individuals and what that can tell us about the geographic range. So here we're looking at a photograph, aerial photography of it's a desert, and I think these are juniper trees. I actually don't remember, but I think they're juniper trees. And so we can see a few things sort of pop out when we start looking at this. And one is that, it's a little bit hard to discern individual trees from this. But you can kind of see that they're not distributed in a uniform way all throughout this image. They're kind of clumped. There's a clump here, and there's a clump here, and then even within here, you can see some clumping, and there's clumps, and then there's distances between them, or areas between them where there's no trees, and then there's another clumps, and that's really characteristic of any geographic range. They tend to be clumpy, and that clumpiness, that grouping sort of pattern, even in itself, is not uniformly spread out. Like the groups are not the same size, they're not located. And what usually tends to happen is let's assume the center of this range is in this area or this direction here. You usually see that towards the center of a geographic range, individuals are spread out in a more uniform way. And as you approach the edge of the geographic range, which we'll assume is in this direction somewhere, you tend to get more grouping, more clumpiness, and more variation. So you get more, large distances where there's nothing, and then, lots of aggregating and grouping together. And so you get lots more variation. And if we look at the actual data, that's exactly what we see as well. So this comes from a study that used, or it wasn't aerial photography, I think they were actually in the field measuring individuals, the density of individuals, so individuals per kilometer, um, over a transect of these palms. So these are for palm trees. And so if we look at this, you can see on the x-axis it's sort of like distance from the center of the range. So all the way over here would be this is the range center, and this would be the edge, the west edge of the range. And what you can see is it's kind of a straight line. Like, yeah, it goes up and down, but you could draw kind of a line through here and get sort of the average for most of this. But then when you get to a certain point, I don't know, maybe that's around 80 kilometers or so, or something, 85 kilometers from the center of the range, then all of a sudden you start to get all this variation. Like it's low, and the density is high and low, and then it drops all the way down, and you get a few over here. So you get some really clumpiness and variation, grouping and variation once you get to the range edge, excuse me, edge of the range. And so we can say individuals tend to be clumped or grouped throughout a species range, with large and frequent clumps towards the range center and fewer and sparser clumps towards the range edges. Something else that's sort of worth mentioning here is that range maps are scale dependent, and that's really a function of the fact that the range itself is scale dependent. The factors that are determining a species geographic range and where a species is located is scale dependent. And so if we have our spatial scale down here, Over here is going to be, these are meters, so this is like 10 meters. It's kind of a small area. You're going to get some microclimate stuff and maybe some dispersal within or between, local populations. And as you move up in scale, you're going to get different factors that are influencing the size and area of the geographic range. You're going to see interactions among species. The habitat and landscape features are going to become more important, and as you move up in scale, your sort of the scales and both spatial and temporal scales, the scales of the factors increase as well. So here you're getting sort of regional climates once you get up to this large scale, long distance dispersal, migration, and things like that. So you're getting a shift in the factors that are influencing as you zoom out, sort of. And we can see that, sort of directly if we look at, here's for an example, the geographic distribution of, I think, an orchid species here in Missouri. Actually, I think it's an orchid. I should have looked up some of what the specific species are. But anyway, we can sort of look at it in different scales here. And so if we zoom out, we can see the pattern of where it's located. But if we start zooming in, you can see some of that clumpiness and that grouping. And if we just keep zooming in until we get to actual like individuals here, you see the patterns change. And the patterns change as a result of the abiotic and biotic factors being different. Maybe it's elevation and I don't know, the slope and aspect and things like that's affecting, where an individual is, where at this scale, maybe it's, the regional climate that influences the size and shape of the geographic range. And so it depends on the scale that you're looking at. So that's always really worth keeping in mind here. Okay, so let's keep moving a little bit and move on from individuals and start talking about populations and species. And there's a couple different things I want to introduce here. So we're going to look at, well, probably throughout this entire course, we're going to see different types of maps, different types of range maps. And I think it's kind of worth introducing what they are and what they're showing. And so we have dot maps, and we have outline maps, and we have contour maps. So dot maps are sort of exactly what you would think they are. They're dots on the map, and they're showing you known locations of where a species has been found. And sometimes, sometimes it's a documented sighting. Sometimes it's a specimen that's been collected for a museum or an herbarium or, a natural history museum, something like that. Sometimes it's a sighting by a researcher or some other, local group or something like that. But they're showing you a verified location. So you can say dot maps show very accurate locations because it was in that location, but they're really also snapshots in time that can only show a small fraction of where a species occurs or where a species has occurred. And so they're not showing it, you might look at this and say, okay, we know it was right there and we know it was right there. Does that mean that it wasn't anywhere in between there? Probably not, but we just don't have the verified information. So and this is going to be the case with all these maps, there's trade-offs. You get a highly accurate location, but you're not getting everywhere that it might be located, and you're sort of just getting this picture in time, one single picture in time. Another type of map is the outline maps, and I think these tend to be the ones that people are more familiar with. They show a boundary line to indicate where the species occurs, but they're highly interpretable. Like you see these in field guides, and anyone has seen it. Really, if you're trying to map the distribution of anything, you probably run into a outline map of some kind where they just draw an outline and this is where it's located. The problem here is that the accuracy really depends on what choices are being made to make this map, because it's not 100%. You can't make it 100%. It'd be impossible to draw 100% accuracy, especially if you're talking about organisms that are mobile. And so you're just sort of taking a best guess. And what you base that guess off of is going to have an influence on the accuracy of it. And so, how careful was the map maker? What kind of data is behind, the outline that's being drawn? How much data is there even available? All these types of things are going to influence the accuracy. And so, while these are really easy to interpret, you just look at that and sort of understand what's going on here. The accuracy can vary, and that doesn't mean that they're not accurate. Sometimes they really are, but it just kind of depends. Sometimes they're combined with dot maps. Sometimes you see these in more academic or scientific journals where they have, some known locations here, and then some of that data is used to draw an outline map. The other type of map is a contour map. And contour maps are used to indicate similar levels of variability to show variation. And usually what we mean here is talking about density, sort of the density of individuals. So, it might be showing a clump here, really high density, these inner circles, and then sort of as you move away from them, you're getting lower levels of density. And you kind of see that when they do the 3D mapping here. And so you're getting more information than a dot or an outline map. You're getting information about the prevalence or how abundant the species is, or how densely populated a species is. But the problem here is that it relies heavily on interpolation to fill in these areas where you don't have data. So, it's not like for, bluebirds or whatever type of species you're doing, and it doesn't really matter for any of these maps. No one has ever gone out and sampled every possible location and found every member of the species and mapped them out. You have some data, and you're using that to sort of form the basis of it, and then you're using interpolation. And we can talk more about what interpolation is later, but basically you're using data from an area that you do have to predict what the value is for an area that you don't have. And so there's like anything, there's trade-offs. Sometimes interpolation can be really accurate if you have good data to predict in other locations, but if you don't have a lot of data, then the interpolation maybe can't be relied on as much. So that's something to keep in mind with contour maps. But we're kind of just on the cusp, or just in the beginning, I would say, of mapping. We're getting lots every day. We're getting so much data all the time, and that allows us to do some really interesting and cool things. Like up here, sort of an example of species distribution maps or species distribution models, which are things that allow us to predict where a species might be. So maybe you can kind of see these white dots in here. Maybe we have known locations of a species, and we can collect information about the environment there. So, how much precipitation, what's the temperature, what's the elevation, what's the topography like, all of it. You can collect as much physical information about that as possible, and you can sort of build a model between the locations, the observance of species and what the environment like is there. And then you can reverse that and say, okay, if we know the environment in other places, we can predict the likelihood that a species will be there. And that's really useful for a number of reasons. Like maybe a species is endangered, or you're worried about it becoming endangered, and you want to know where it might be, and you can find locations of it, or predict potential locations of it in places where you may not have known. Another area where this is really useful is climate change. So if we want to know what the climate is going, if we have some idea of what's going to be happening in the climate, or we want to look at different potential scenarios of climate change, we can sort of predict out into the future and say, okay, if conditions are like this, where might that species go based on, where it's located now. We can map movement and migration patterns to a crazy level of detail. Not that we know everything, we don't know everything, but it's kind of an exciting time for that type of science. And we can collect genetic and genomic data and look at sort of phylogeography, where we're looking at the genetic information on a spatial scale and get information there. So we'll talk more about these as we go through the class, but it's sort of, I'd say we're just on the frontier of what we can do as far as mapping species distributions. And that's a pretty good segue into, if you're working your way through the module here, I'm going to end this video, and then there's a reading about drones and remote sensing that can be used for mapping, which are just another area of sort of this frontier of GIS and mapping for biogeography. So keep working your way through the video, and I'll see you in the, or through the module. I will see you in the next video.